In the last video, we introduced the concept of passive transport, wherein a molecule or solute goes from an area of high concentration to an area of low concentration, which we also call moving down its gradient. However, we have a couple of specialized types of passive transport that I want to go through now. So let's imagine the following situation. We know that cell membranes are selectively permeable, meaning that some things can cross and some things can't. So we could very easily have a situation where we have a cell surrounded by a membrane. We have some solute that can't get across the membrane, highly concentrated on one side, with a very low concentration on the other side of the membrane. That molecule, however, still wants to be equally spread out, still wants to reach equilibrium across the membrane, even though it can't move directly across. So in this case, we're looking at a cell in a hypertonic solution. So here we see our membrane. Here we see a solute dissolved outside the membrane in a high concentration. Here we see a solute dissolved inside the cell with a low concentration. So let's say that this solute is unable to cross the membrane. How can we make it so that we end up with an isotonic solution, so that we end up with the solute evenly distributed on each side of the membrane? This brings us to our first special case of passive diffusion. So one thing that can happen in this situation is that instead of having the solute itself move, instead water diffuses across the membrane. So the cell membrane is permeable to water. It's actually pretty highly permeable to water for a number of reasons. And so when we have a situation where there is a substance dissolved in water, and there's a difference in the concentration of that substance on one side than on the other side, that water can move to basically water down the area with a high concentration of whatever it happens to be. The other way we can flip that idea and work on it, the other way we can flip that idea and look at the other side of it is that in a situation where we have a lot of this red molecule outside the cell, we actually have less water here. So we have a low concentration of water where we have a high concentration of solute. And so inside the cell, we have a high concentration of water where we have a low concentration of solute. So they're kind of the inverse of each other. So in this case, in our hypertonic solution, we're going to have water move from the inside of the cell out of the cell to water down this highly concentrated solute outside the cell. And this is called osmosis, when we have the diffusion of water across a membrane. Now, in animal cells, this is going to do specific things to the shape and structure of that cell. The first one is in that example that we just looked at, our hypertonic solution, where we have a solute concentrated outside the cell and water moves out of the cell to go water down that environment. What happens to the inside of the cell is that suddenly the inside of the cell has less water, which means it has less volume. So that cell is going to shrink because the cytoplasm is mostly water. And we call this crenation, when a cell shrinks and gets all crinkly and lumpy because suddenly it kind of has too much skin for the amount of volume inside the cell. And we can have the opposite happen, and the opposite will happen in a hypotonic solution. So if we have only a little bit of a solute outside a cell, we have a lot of water outside the cell. And on the inside of the cell, in that case, we have a lot of solute, but only a little bit of water. We're going to have water move into the cell. It'll move into the cell, sometimes so quickly and so much, depending on those concentrations, that it can actually swell to bursting. So we call this osmotic lysis, when a cell takes in so much water that it totally bursts from the increase in volume. In plant cells, this works a little differently because plant cells have cell walls surrounding the outside of the membrane. So a plant cell will still have a cell membrane, but on the outside, it has a structure, kind of giving it extra support. So instead of undergoing crenation and getting all knobbly when water moves out of the cell in a hypertonic solution, the walls of that cell just kind of cave in a little bit, which we call plasmolysis, which is basically when you see a plant wilt. The opposite is turgor pressure. So in a hypotonic solution, so if you think of like pure water, that water moves into the cell to water down all the proteins and stuff inside the cell and fills up the volume of that cell. So it gets bigger and bigger, the volume inside. But because these cell walls are more rigid and stronger than a membrane on its own, instead of bursting, they just get pressed outward. And so that cell becomes really firm. So like the plant crisps up, like the opposite of wilting. So that's what happens when you water your wilting plants. The other thing that can happen if we have a solute that is unable to get across our lipid bilayer is that we can find an integral protein, so a protein that goes all the way across the membrane, that's able to carry that solute through it. We call this facilitated passive transport. So if we look at a situation and we can see that a molecule is going from an area of high concentration to an area of low concentration, we know that's our favorable direction, won't require energy, passive transport. If it can't dissolve directly across the plasma membrane, it's going to need a protein to be present. So we're going to call that facilitated passive transport. And we're going to contrast it with simple diffusion, which is what would happen with things like oxygen that can just diffuse directly across. But otherwise, this is going to work really similarly. So let's take a look here. So facilitated passive transport is mostly going to happen with ions and also some small polar molecules. So we see here we have an integral protein, which is a protein that goes all the way across the membrane. So there's an outside part and an inside part. We remember that that happens because proteins have this complex shape where different regions can have different chemical properties. So all that happens in facilitated passive transport is that our solute goes from an area of high concentration to an area of low concentration. But it does that by going through the protein. So here we can see that this protein has a tunnel or a channel going through it. And we can see that this highly concentrated yellow molecule, so there are more of it here on the outside, go down their gradient, so from high to low concentration, by moving through this channel in this protein. So here's a comparison of our different types of transport. So we talked about diffusion. So simple diffusion is for things like oxygen that can just dissolve directly across the membrane because they're nonpolar, so they can get through these nonpolar fatty acid tails. Now we have facilitated diffusion, where we'll go through our protein still from an area of high concentration to an area of low concentration. And in our next video, we'll talk about active transport, which we're going to contrast with passive transport because instead of going from high concentration to low concentration, we're actually going to go from low concentration to high concentration. Before we get there, I want to take a second talking a little bit more about those integral proteins that our different ions and molecules are able to travel through. So there are basically two types: channel proteins and transporters. So a channel protein is what we saw in that previous example. So in a channel protein, you have a transmembrane protein, so going all the way through the lipid bilayer. It's got a passageway down the center and things can just diffuse through that passageway. So it no longer matters whether they're polar or non-polar, because there's like this hole, this gap, where they don't have to interact with the lipid bilayer. Many channel proteins are gated, which basically means that you can like turn them on and off. We're not going to worry about gating in this unit. And if you see a channel protein, it'll really be indicated because you'll see this direct passageway all the way through. Now we contrast our channel proteins with something called transporters or carrier proteins. Now, they work a little differently. They're still able to move molecules from one side of the membrane to the other side of the membrane. But instead of just forming an open channel, they're going to actually bind to the molecule on one side and then change shape to kind of like push it through to the opposite side. That can be a passive process or an active process. So it sometimes will require energy, sometimes won't require energy. So you still just want to focus on the gradient. So it will not require energy to do that shape change if we're going from an area of high concentration to an area of low concentration, but we are going to need energy if we're going from low concentration to high concentration. So our transporters or carrier proteins are sometimes going to be doing passive transport, sometimes they're going to be doing active transport. The channel proteins can only do passive transport because they can't control which direction those molecules are going at all. So they're always going to go in the favorable direction. Our transporters mostly move things like sugars, amino acids, and nucleotides. And we have 3 types of transporter, and we name them just by basically the direction molecules are moving through them. The simplest is a uniporter. So in a uniporter, we have one molecule go one way through our transporter. That's it. In a symporter or a co-transporter, we're going to be moving 2 molecules, 2 different molecules, or an ion and some molecule across the membrane, and they're both going to move in the same direction. So if they're going in the same direction, it's a symporter or a co-transporter. If they're going in opposite directions, it is an antiporter because they're going opposite to each other. So one molecule is going this way, one molecule is going that way. That's an antiporter.